Thanks a lot. I'd like to start off by thanking the organizers for the opportunity to be here. I earned my PhD in the green and white building right next door. And as a young aspiring theorist, there was a great deal of mystique in walking by this building that was filled with nothing but chalkboards. So it was quite literally a dream of 25 years to have the opportunity to be invited and participate in a workshop here. So I'd like to start off by, the theme of the talk is structure function, and so I'd like to start off thinking about this on the most abstract level. If you asked me to identify the single most important concept in biology, well, I'd have to say evolution. But if you asked me the second most important concept in biology, I would go with structure function. And of course, this is a concept that propagates across scales, from hydrogen bonds and Watson-Crick base pairs to the folds of a mitochondria, all the way up to the shapes of the beaks on Darwin's finches. But for a room full of molecular biophysicists, perhaps the most familiar example of structure function is that of a globular protein or an enzyme. Okay? Now this is of course an incredibly complicated system, but one we understand very well. But in order to understand it, we require these structure function concepts propagating over a huge range in scales. So let me demonstrate that to you. At the largest scale, the way we think about enzyme is the specific positioning of enzymatic or of groups. I've got a specific binding pocket, and of course I've got some catalytic residues that do the work. And this positioning is enabled by the fold of the protein, which is generally driven by the collapse of some sort of hydrophobic core. Okay, now this positioning in turn is determined by the amino acids that come into the system. And the characteristics of the amino acid that drive this folding are characteristics like hydrophobicity and polarity and charge. Okay? Now, these functional characteristics of the enzyme or the amino acids in turn come from the chemical structure of those amino acids. And that chemical structure is determined by things like the bonds and the electronegativities and the valence of these electrons. These characteristics in turn come from the shell structure of atoms. Okay? And in turn, the shell structure of atoms comes from the solutions of the Schrödinger equation in spherical coordinates. Okay? Now, the point I'm trying to make here is that in order to understand how an enzyme works, we require structure function concepts ranging over this entire range of scales, from the angstrom up to a few nanometers for an enzyme. And I've only covered about 1.5 decades in length scales here, and yet I need at least 5 different concepts in order to really understand how an enzyme works. Okay, so this is my positive control. I've taken a complicated system here, and we understand it very well. So now let's do the negative control. Let's try and understand this complicated system using the wrong concepts. So what I'm going to do here is I'm going to step down 1 step in scale. I'm going to go from the fold scale to the amino acid scale. And I'm going to say, how can we understand how this enzyme works? And what I get is an amino acid connectivity map that is completely and utterly useless. Okay? The catalytic residues on this, highlighted in red, are not even connected on this map. Okay? So the implication here is that if we apply concepts that are ill-suited for the scale that we are interested in, what we end up with is complexity. Okay? And what we need is instead is the concepts that are appropriate at the correct scale. Okay, so these are the concepts I've talked about before, and we understand things going from the electron scale to the enzymatic scale very well. I would argue if we keep pushing up in scale, I need to add one more to the list, and that one is molecular feedback. But once we get above this, we've got almost nothing in cellular biology until we get all the way up to the scale of the organelles. Okay? And these are the concepts that my, 8th graders and 11th graders come home and ask me about when they're taking biology in class. Mitochondria is the powerhouse of the cell. The nucleus stores information, that sort of thing. Okay? And so our challenge here is to identify these concepts that allow understanding of these complex systems at the supramolecular and above scale. And the implication is that concepts like charge and hydrophobicity that have proven so useful and so powerful at this scale here might be unnecessarily unwieldy for us when we move into the supramolecular. So we need to be prepared to leave those concepts behind if the situation calls for it. Now, the other point I want to make is that this hierarchy of concepts that I've outlined here are all emergent properties. And I don't need to belabor that point. Everybody in this room here is here because they are interested in emergent properties. Tanya gave a nice introduction to this the other day. Okay, but as a few comments on this, I'll note that that term has different meanings depending on the communities you talk to. So I went digging into biology textbooks, and when I see the term emergent properties come up, what they're usually talking about is hierarchical structures. Okay? In the physics community, and largely for most of the people in this room, I would say that we use the term slightly differently, and to borrow the words of Anderson, our definition is more is different. In other words, emergent properties are those characteristics that we cannot expect by looking at the components of the system, okay? And so because of that, if we want to push this conceptual hierarchy further, we are going to need top-down methods, both theory and experiments, and we've already seen some fantastic examples of this over the last day and a half. So what I'm going to do over the remainder of the talk is I'm going to take Anderson's advice perhaps a bit too literally, and instead of talking about a particular system that we have studied, we're going to talk about an ensemble of systems that we have studied over the last 5 to 7 years here. And what I'd like to do here is extract some emerging themes that come from this work. And of course, this is highly preliminary, and you know, this is very likely going to change over the coming years here. But what I'm going to do here is talk about three themes that seem to be popping up over and over again: internal state, density as a mechanism for specificity, and the importance of small effects. And at the very end of this, I'm going to come up and I'm going to propose, again, a very preliminary proposal for what structure function at the supramolecular and condensate scale might look like. Okay, so the first theme, and this is, if you take one thing from my talk, this should be the one, is that condensates have distinct internal states. Okay, we have seen this over and over again in these systems that we have investigated top down. The first example is Tanya's spot DAX condensates. It had this very interesting phase diagram that consisted of both gel states and liquid states, and we showed that you could change between that stoichiometrically. About a year later, we looked at Mike Rosen's Sumo Sim condensates, and we found that an emergent property of the condensate was that they had the ability to specifically bind or reject very closely related molecular clients, and you could tune between those different recognition modes. We've seen this in unpublished results on DNA binding proteins, but the story I want to take a little bit more time on is this story about viral condensates. Now, this is the experimental work coming out of our Alexander Borodovka's group. He's interested in rotaviruses, and the two key molecular players that are going to show up here are NSP2, which is an RNA chaperone. It's got a globular folded structure that is donut-shaped. It's a higher order oligomer, and it's got a lot of positive charge because part of its function is to bind RNA. The other molecule involved is NSP5. It's primarily disordered, although it has an oligomerization domain at the C-terminus, probably mediated by coiled-coil interactions or something of that flavor. So what Alex's group did in collaboration with Thomas right here is they collect phase diagrams of these two proteins. This is mixing NSP2 and NSP5 together, and you see the following phase diagram. Now, the next thing they did, and then they take these two proteins, they label them, look at them in infected cells, and you know, reassuringly, you see qualitatively the same behavior, the formation of puncta containing these two proteins. So the next thing they did is they took NSP5 and they engineered the sequence of it to take it from a sequence that had high phase separation propensity to a sequence that had very low phase separation propensity. And sure enough, they take this engineered variant, look at it in vitro, and there's no phase separation. So they take this engineered variant, they put it into a virus, use it to infect cells, and they still get condensates. Okay, so this is the puzzle, okay? Why is it that this thing is not forming condensates in vitro, but it is forming condensates in cells? Now, the clue here comes from the shape of the phase diagrams, okay? So this is the phase diagram for the wild type NSP5 molecule, but again mixed with NSP2. And the feature that I want to draw your attention to is just how straight these phase boundaries are. Okay, if I put those dotted lines in, you can see that these things are very straight. There's very little in the way of reentrant behavior going on here. Okay, however, they take the phosphorylated version of the same protein, and now you see these very distinctive curved phase boundaries indicating that there is reentrant behavior going on. Okay, and so what that says to me is that something has changed in the mechanism of interaction. Okay, we've gone to some type of interaction that cannot saturate. Okay, something like FUS does not saturate when you take it to ultra high concentrations, whereas down here you've got some sort of saturating interaction here. So how can we take the molecular architectures of these two molecules and get models that have each of these two characteristics? Well, this is my non-saturating model here. We're going to use NSP2 as a hub, and I'm going to start assembling some NSP5s along it. And I'm going to have it bind via the C-terminus here, which is, again, the oligomerization domain. And so we've got this phase-separating IDR at the end here, and so these little pinwheels are going to phase separate into some sort of fluid. And if I have a stoichiometric excess of NSP5, this thing is just going to happily intercalate into this fluid, and there's not going to be any significant disruption going on, and so I still phase separate. Okay, in order to build a saturating model, I'm just going to invert the geometry. I'm now going to attach the NSP5 via its N-terminus, and so now I've got these oligomerization domains dangling off, and so when this thing phase separates, we've got these sort of specific oligomerization activities that are going to link it together into the dense phase. So the question is, how can you get a conversion between these? And it turns out that the phosphorylation sites on NSP5 are predominantly here at the N-terminus. So if I phosphorylate this molecule here, then I've got this excess of negative charge here, and so now this negative charge here can compete for interaction with the positive charge on the RNA chaperone and the hub. And so this molecule is going to invert its geometry. Okay, now, since proposing this particular mechanism, Alex has done lots of experiments in order to verify what's going on, and I'm not going to go through those, but what I am going to do is show you what I believe to be the really convincing one, and that's hydrogen-deuterium exchange of the binding between NSP2 and NSP5. And what you can see is that the non-phosphorylated version, the primary protected regime is here at the C-terminus. However, when they look at the phosphorylated version here, all of a sudden the primary binding mode jumps over here to the N-terminus, and refreshingly, that binding is right in the middle of these phosphorylation sites that are labeled with the blue lines here. And so what happens? When I now oversaturate this phosphorylated version, now I bind up these oligomerization domains, but now I have these IDRs whose phase separation propensity is going to be much weaker because now I've coated them with a bunch of negative charge, and so this is no longer going to have the ability to phase separate, or at least much more weakly. So what this means, what this suggests, is that these condensates are actually changing their conformation over the life cycle of the virus. Okay, when I look at early infection, whether I'm looking at the wild type variant or the low phase separation variant, I see a big difference in the amount of condensates that form. That's quantified up here. And this is due to the fact that they are forming by these IDR-IDR interactions here that have been engineered to be weak down here. However, later in the infection life cycle, after the host kinases have had a chance to kick in, we now revert to this alternate conformation driven by these stoichiometric interactions. Okay, the second theme is that sticker density has shown up for us in numerous models here as a mechanism for selectivity in condensates. Okay, now I want to be clear here. I'm not making any claim at all that sticker density is unique as a selectivity mechanism. We're going to see, we saw an excellent talk from Will. Ned's going to talk about selectivity later on. This is just something that has showed up in a few of our models here, and so I would put this in the category of curiosity at this point in time. Okay, this first showed up in our sumo sim condensates, where we saw this ability to select between different clients. Later on, we saw it in Tanya's SPOT-DAX system, which has this interesting correlation between both the biological activity and the condensate formation activity that is consistent with a density type of mechanism. But the one I want to spend a little bit of time talking about is these polyubiquitin condensates formed in the presence of shuttle proteins. So as most of you in this room are sure aware, polyubiquitin chains are used to target proteins to proteostasis or protein quality control targets. But the specific protein quality control outcome seems to be determined by the particular linkages depending on which side chains are used in order to ligate these ubiquitin molecules. So this was the question that Carlos Castaneda was interested in. He was studying this protein called ubiquilin 2, which is an intrinsically disordered protein as illustrated there, but in all the cartoons I'm going to show you, I illustrate it as a red oval. Okay, now ubiquilin has the ability to phase separate using homotypic interactions by itself, but it also has the ability to bind ubiquitin with a one-to-one stoichiometry. Okay, so what Carlos did is he used various polyubiquitin constructs and found that there was this uniform trend that the longer the linkages were between these chains, the more favorable it was for phase separation. So then he wanted more control over these linker lengths there, so he went to this sort of branched architecture, and instead he found the opposite result, that as he made the linkers longer, it became less capable of phase separating. And so this is what we set out trying to understand here. And what we did is we built a model that had the following two characteristics. The first characteristic is that if I want to insert an ubiquitin module into this ubiquilin fluid, there is going to be some disruption effect, okay? And so this is going to come with some sort of repulsive energy. On the other hand, if I have a polyubiquitin chain, then I get this enhanced valency effect, and this construct is going to have a higher net driving force for phase separation than an individual ubiquilin. And this model does a pretty fantastic job of describing the experiments that what he was seeing. So what this model does is it predicts that there is a relationship between the geometry of these ubiquitin chains and the ubiquilin fluid. Okay? And that if you have a ubiquitin chain that has an appropriate spacing, a spacing that is commensurate with the mean density of these ubiquilin molecules within their fluid, it's going to fit well. Whereas if the molecule is too small or too large, you're going to get a local disruption of the network. And so the prediction there is that if Carlos can engineer a linear molecule that has dimensions commensurate with this, then all of these molecules should fall on a common curve, which is exactly what he found. And so this model that this suggests is that these shuttle proteins are reading these different polyubiquitin linkages via the emergent properties of the condensed phase. It's this spacing of the condensed phase that is more sensitive to phase separation depending on the polyubiquitin chain that you use. And I'll take this moment to put in a teaser that next Wednesday, for those of you that are around, using this sort of spacer density as a mechanism for selectivity is one of the topics I'd like to talk about. The third theme here is that we are looking for small effects, okay? Small perturbations have a big effect, okay? And I'll start with this with a story. This is, again, goes back to the SPOT-DAX story that was doing in collaboration with Tanya. And we came up with a model that, had this, reentrant behavior between the dilute and the dense phases. And, I felt like this was going to behave right. And so then Tanya and her postdoc Jill measured the two parameters in the model, which were divalent binding and monovalent binding. And they sent me these numbers, and I took one look at it and said, well, back to the drawing board, this is never going to work. Okay? But in fact, as soon as we plugged those parameters in the model, it nailed the phase diagram just right. Okay? We've seen the same sort of thing pop up over and over again. We see these DNA titration curves here, just these tiny little perturbations in the length of the DNA give qualitatively different shapes in the titration curves. These binding parameters here are on the order of fractions of a kT per ubiquitin module here. But what I want to argue is that there's reasons that this has to be true, okay? This isn't just an accident of our models here. The first is that the very nature of emergent properties means that the interactions that are small on one scale suddenly become large as you move up in scale. Okay? The second reason is biologically you want to have tunability and variability and sensitivity. So, this is allosteric regulation. I need to have these states that are convertible on a scale of kT or so, and something similar is going to happen when you're talking on condensates, but again, you've got lots of interactions that are going to add together and give you some very big barrier unless you're talking about something small. Third, we are looking at a competition between very similar states, okay? So this is an example here. This molecule in a homodimer is in a very similar chemical environment to these molecules that are immersed in the dense phase. And there's very small differences in the free energies between these different phases. And so so we're looking at very small changes from one state to the other. And the fourth reason is our old friend, entropy-enthalpy compensation. And we're familiar with this in the context of things like the hydrophobic effect and protein folding, but it shows up in a big way here. So we've seen this again in these DNA histone binding interactions where we just had this massive compensation between polymer entropy and electrostatics. Again, this spacer story that I teased before, we see a cancellation between two absolutely huge entropic terms. And then what we find is that you can actually tune this enthalpy-entropy compensation, particularly at interfaces, and get very different interactions at the interface than you have inside the bulk. So what I want to do here is now return to this question that I laid out at the beginning. What does structure function look like on the scale of condensates? Okay, now in order to address this question, I've been borrowing a lot from traditional hard condensed matter. And this is a place where I think the theoretical community has a lot to offer here. So what did they do in hard condensed matter? Well, what they did is in these systems, they identified order parameters. Okay? And what do I mean by that? In my usage of the term, an order parameter is that collective variable that you need in order to connect some microscopic mechanism to a macroscopic phenomenology, whatever collective variable that you might need. And to give you examples from traditional condensed matter, in superconductivity theory, there's a band gap that plays a role in the Peierls instability. It's a lattice distortion. And of course, in our old friend, liquid-liquid phase separation, it is a density order parameter. Okay? And these similar order parameters have popped out of each one of our toy models, our analytic models for all of these. In some cases, they're parameters that we put in by hand. And in other cases, there are parameters that emerge from the theory after it's done. Okay, so with that, I want to summarize what I've talked about. We've seen three recurring themes in the still relatively small number of systems that we've looked at. One is this idea of switchable conformational states. Two, this curiosity that the density of stickers seems to be a recurring mechanism for selectivity. And I don't know, maybe that's just because this is the mechanism that we haven't expected that it's popping out, but again, I think this is more of a curiosity at this stage. And then the third theme is that we are looking for very small effects, okay? These transitions between different states are going to be absolutely tiny, especially when you look at them on the molecular scale, and it's not until you add them up and get collective behaviors coming out that these effects add up to something significant. And then finally, my preliminary proposal for how we describe this is using order parameters in the same sense that they came out in traditional condensed matter in order to describe this connection between microscopic phenomena and macroscopic. So with that, I'd like to put up my thanks. This is all of this is collaborative work with many of the people that are in this room. I'd be happy to take any questions.
Fantastic talk, Jeremy. Question on your point 3.
Yeah.
I want you to speculate.
Sure.
The point 3 was small perturbations have big effects, And in the lens of evolution, big effects means you generate variation, but the flip side is you also need to maintain robustness. And in the information theoretic sense, that's a very stiff mode that you investigated, but surely there are other sloppy modes to the system. So can you speculate on what you think are the stiff and sloppy modes for condensate?
Well, I guess what I should say is that if we're looking at molecular interactions, you're going to see tiny little differences in the states. But when you put them together and get an emergent property, all of a sudden now you see things that are big. So another way of looking at this is imagine doing some sort of self-assembly process here where you're building small components, making intermediate components, and making then larger components still. Okay? At every scale in that, you need to have reversibility. So I want my small components to have order KT binding energy so that they can try, and break a few different interactions. But then I also want my intermediate building blocks to have that order KT binding interaction so their reversibility, okay? So that intermediate building block is forming lots of the small molecular interactions, which means that those have to be much weaker in order to give me that reversibility on that scale. So when we're talking about biological information content here, we need to have these states that are switchable on the energy scales that are useful. And that's, say maybe ATP scales or maybe something larger than thermal. But going up to these large supramolecular scales, that corresponds to very weak interactions at the molecular scale. Does that answer your question?
Not really, but I'll ask afterwards.
Okay.
So there was a question there first, and I'll come back to you.
So your observation about the 2D phase diagram being either parabolar or hyperbolar in terms of searchable and unsearchable interactions. That's very insightful. I've actually been positive about this for a long time. So, but even it's a matter of degree, isn't it? That there's nothing really unsearchable, right? If you go to really, really high concentrations in either arm, it's going to curve up eventually, right?
Yeah, and you can see that on the phase diagrams I put there. There was a tiny little hook at the end. Yeah.
Right.
Yeah.
Yeah. So your second theme, isn't it? The simplest case is simply single polymer solvent that's considered by Floyd-Huggins, right? The critical temperature for a single polymer phase separation scales with the length of the polymer chain.
Fully agree. Yeah.
Okay.
Yeah. No, so from that perspective, that's a very trivial observation, conclusion. I totally agree with that.
Okay.
Yeah.
Next question here on the left.
Great talk, Jeremy. I have a question regarding those order parameters that you alluded to.
Yeah.
So what are your thoughts of what are they and how can we get into them?
I don't know. I mean, I think we're sort of, at the scale of, we've got that first crystal structure of myoglobin and trying to ask, how does a protein work? We need to have more examples of it before the trends start to come out. So, I mean, all I know is that in the models that I've worked out here, I'm not seeing common themes, pop out. But this is also true for, folded protein. I mean, you've got enzymes, you've got structural proteins, you've got information transporting proteins, and trying to draw, common themes about how each one of those works. You need a bigger data set than this. So we need a PDB of condensate structures before we can really start to answer that question.
This might be philosophical, though.
Bring it on. Yeah.
Okay. No, it's just because you mentioned the PDB. I mean, so with AlphaFold, for example, again, there are different proteins. We don't necessarily know how AlphaFold gets it, but it does get it, right? So with condensates, do you think that's the same? Do you think that there are perhaps there is information in the sequence or that is there, but we just don't know how to reach it yet?
Well, ultimately, it has to be in there. Yeah. So I don't know. It's a good question. At one point, I started thinking of myself as a structural biologist, just on this scale. But I still don't self-identify as a structural biologist.
All right, if there's any quick last question.
So I'm very curious about your H1 DNA interactions, where you said very small changes in DNA length give
Yeah.
very different ITC results. Can you allude a little more on how this emerges?
Okay, the short version of the story, and there's a reason that I didn't give a talk on that. It still shows up as unpublished, so the story isn't done right now. But we think that the essential ingredients have to be that there is a cooperativity between nearest neighbor DNA molecules and their H1 binding. And this plays a huge role in the small oligomer phase, okay? So when you start talking about heterodimers versus trimers and tetramers and that sort of thing, whereas it does not play a role in the dense phase. And so that tiny difference, you know, because again, a ligament in this phase looks a lot like a dense phase. You've got very similar types of electrostatic interactions, but just tiny little tweaks in the stability of that oligomer just lead to huge effects in the stability of the apparent stability of the condensate. Yeah, but we can talk about that more. I'd show you. There's a lot more to say there. Yeah.
All right, then let's thank the speaker again.